open this computer. I'm going to broadcast. I'm going to share. Welcome, everybody. For those of you who are just joining us, we're glad that you can be with us today. We're going to ask that you open up your chat box. Please let us know who you are, where you're coming from. Just make sure to hit all panelists and attendees. That way everybody can see who's joining us today. We like to have a nice community here. So if you just hover your mouse towards the bottom of your screen, you'll find the menu where you can open up your chat box. And again, great. I see some of you are finding that. Welcome, Boone and Cannon. Welcome, Grant from Chicago, and Amanda and Wisteria. Recognizing a lot of these names. Nice to see you all again. Oh, we got Luke from New Jersey and Haley from Maine. I always love seeing Mainers because that's where I grew up. So shout out to anyone from Maine who's with us today. We have Emily from Lebanon and Land and Quincy and Anna. Oh, welcome everybody. It's so great to have you all join us today. So if you're just coming in, find the chat box. You hover your mouse towards the bottom of your screen and just introduce yourself, your name and where you're coming from today. We're just going to give it a couple of minutes because we are expecting a fairly large crowd today. So we want to give everyone an opportunity to come on in. If you need closed captioning today, I am going to put the information in the chat box right now. It's also at the bottom of the screen for those of you who might need those services. But keep introducing yourselves. I love seeing Evelyn wants us to take, she's an alien. Take me to your leader. Okay. No, so someone asked, can you see or hear us? No. In our science cafes, the way we have these set up, because we use a webinar format, you can see and hear me and our presenter. But the only way we can communicate with you is you all have to write in the chat box. We can't see you, we can't hear you. You don't have those features. Love this. Welcome everybody who's still introducing themselves. So I'm gonna ask you to keep introducing yourselves while I go ahead, and I just wanna go over some of our protocols for when we're in this virtual space. So as I just mentioned, you all are muted. You're not allowed to unmute yourself. That's not a feature today. So we're gonna ask you to share in the chat box. The chat box is gonna be used just for sharing thoughts on today's topic. So our presenter might ask you a question and you would type your answer in the chat box. The chat box really is not meant to be chit chat. We find that to be distracting, and it does distract other participants. So we do ask that you use the chat box only as it relates to what's happening with the presentation, and especially if our presenter asks you for some comment or feedback. There is another option, and if you look at your menu at the bottom of your screen, you're gonna see there's a Q&A box. That is where we're gonna put all of the questions for our presenter today, because the chat box tends to lose things in there. But if you put your questions in the Q&A box, those stay there until we answer them. Now I want you to know, sometimes the questions might build up. Our presenter will find appropriate times to stop and answer your questions, but some may not get answered to the end of his presentation. But if I see a question that might need to get asked earlier, I'll make sure to let him know, okay? So we ask that you all just be courteous and respectful today. And again, a lot of that has to do with the chat box. We just don't want you to create any distractions. We have found that if the chat box becomes too off topic or too distracting, then I will have to just take the privilege away. And I don't like to do that because we do use the chat box as part of the learning. So we ask that you stay engaged and participate fully today. And with that, I just want to now go ahead and introduce our presenter. We have Huy Tu from the UVM Vaccine Center. And I'm just gonna read you Hugh's, I always say Hugh, Huy's bio. Huy is a PhD student at the UVM Vaccine Testing Center, which is a division of the Microbiology and Molecular Genetics Department. His research focuses on how human develops defense and memory against dengue and Zika infections. These works contribute to our understanding of protective immunity to these viral infections and can be applied to the development of immunization strategies against these viruses. So I want to take this opportunity to welcome Huy, and I'm going to stop sharing my screen and I'm going to turn it over to Huy, and I'm excited to learn all about pandemics, virology, and immunology.
All right, let's see. Thank you, Lauren. Hi, everyone. Thanks for tuning in today. All right, let's see. Do you see my screen?
Yes, you just need to go into your slideshow view.
Okay, slideshow. Come on, there you go. Now I need to.
Great.
All right. Oh, good. Okay, so thank you. So I am super excited that Lauren asked me to come back to talk with this group about what I study. And many of you might have come to the workshop that we did in November about how to combat infectious disease. And I didn't plan it, but the month after we had an outbreak that lasts to today. And so now that we are in the middle and maybe hopefully at the end of the outbreak, I thought it would be appropriate to talk a little bit more about the fundamentals, the basics of what I study, as opposed to going too much into details of, you know, Zika infection and that sort of things. So today we are looking at virology. So we're gonna go through some basics about what a virus is, you know, the biology of a virus. And then we'll move on to immunology, which focuses on understanding how the human body mounts a defense against infections. And in this case, we're dealing with a virus infection, right? Coronavirus. So the first thing I want to do is there's a poll number 1, and this is it's not of great importance, but this is something fun that we always like to bring up, you know, at any virology class, or even if you go to a virology conference, you know, this is something that people toss around quite a bit. And the question is virus alive, right? So so I'm not gonna go into, you know, the details of like what the correct answer is, because this is a very contentious topic, and the answer always seems to be split between split 50-50 really, if you ask any expert in the field. And I hope, you know, at the end of this portion today, you may be able to come up with an opinion for yourself as to why it is alive or if it's not, and if you are able to justify your answer, because that's really how science operates. You give an answer, you need evidence to back up your answer, right?
And I'll say the poll was launched. If for some reason you didn't see it, because for some reason not everyone can see when we launch polls, you can always put your answer in the chat box, okay? But I'm going to end this poll now, and you can see that just like Huy said, it was almost split. A little bit more on the yes than the no.
All right, awesome. Yeah, so that's the answer that we usually see, right? And so so what is a virus? Hold on, where am I? what is a virus, right? So here I have several images of viruses, and some of these might look familiar to you and some maybe not. But these are the images of influenza virus, so your seasonal flu there. The corona, which is very popular these days, and the reason why it's named corona is because of all of the spikes around the perimeters of the virus, right? That create, the image of a crown or a halo, and that's what corona means. And then you have the very distinctive looking Ebola virus, which just creeps me out every time I see it, but it makes for a very good effect. So virus is classified as a microbe, right? What it means is a tiny biological agent. And I have a, website right here, which I'm not gonna go into since I've got to like switch back and forth between different screens, and that just creates more issue with my terrible internet. But you can go to that website scale, and I have it at the end for you. And I'm sure Lauren can send out the information to all of you. You can see, how big the viruses is compared to, your, say, you start at a coffee beans and then you go to a grain of rice, and you keep zooming in, and you will see, like for example, your skin cells is about this big, and then E. coli cell is tiny bit, and then the virus is even smaller than that, right? So virus is one of the smallest microbial agents that we know of, right? So one, it's tiny, it's a micro. Two, it's a obligate intracellular agent, and it's a fancy term for the virus being dependent on the host cells, right? So as with any biological creatures, the goal is to pass on your genes, your genetics, right? And it's the same with the virus. The virus needs to pass on its gene, even if you consider it alive or not. It's not thinking, I need to pass on my gene. That's just what it does, right? So it needs to be inside a host cell in order to pass on its gene. It needs to be inside a cell in order to reproduce, right? And so that's why we call it an intracellular microbe. And it's obligated to be in there. And so that's why it's called obligate. So those are the three items that I wanted to talk about when we think about what is a virus. It's different from a bacteria or bacterium or a parasitic worm, for example, due to size and due to what it can do inside our cells. Many bacteria are not required to go inside our cells in order to reproduce, right? And so let's look at the poll number 2. So I just told you that the virus need to be inside the host cells to reproduce. And to reproduce or to make more viruses, it's gonna need material and the cellular machinery to make all these new viruses. So the question for you is, which of these components come from the host cell? You have viral proteins, which are building blocks for the virus. You have the viral genome, which is the genetic material for the virus. And some viruses are covered by a lipid membrane, which is a fat. It's like a cell membrane. In this case, it covers the virus. So, and then all of these items above are coming from the host cell. So let me know what you think.
All right, I'm launching the poll. If you see it, take it. If not, type your answer into the chat.
Right, and while you're working with the poll, I see a question. Is it possible to engineer a virus that combats other viruses? very interesting thoughts. You know, I don't think we can do it at this point, because each virus interact really, the interaction is between the individual virus and the host cell. There has been no study to see, you know, if this one virus can interact with another virus, because if they're by themselves, they're not really do anything, right? They're just like a block of genetic material and protein. When they're in the cell, that's a little bit tricky to study, because when one cell is infected with one virus, the virus is able to sort of like change the behavior of that cell to make it less or resistant to further infection. So this is something interesting that we have not looked at. But you know, maybe one of you can pursue this in the future. All right, so now all of the above wins out, and I agree. I agree. Good job, everyone. Yep. So a virus needs to be in the cell, and it needs the resources, the material, to make new viruses, and all of those materials are provided by the host cells, right? So it's just smooching off the cells. Okay. So now let's look at the structure of the virus. So here I want to talk about the components, what makes a virus, right? So here is an image of the influenza virus, and you can see a gray background, and then there are a ton of little, you know, spots on this gray background. And if you slice this in half, let's see, right? Cut it in half, like cracking open a coconut, you see a little bit more complexity there. So the gray background on the outside is now a gray layer. That's actually a lipid layer, right? It's a membrane that when the flu virus infects a cell, when it gets released from that cell, it takes away the lipid layer of that host cells, right? And then next, we have a yellow layer, and that's the matrix protein, right? So these are usually considered structure protein because they form the structure of the virus. They are the infrastructure, they form the support of the viral particle. And then in here, the coil, what you're looking at are genetic material, right? So genetic material can be DNA, it can be RNA. Those are different kinds of molecules that can carry instruction on how to pass on this virus, this gene. And usually the genetic material is packaged with other proteins, right? So they're not just like loosey-goosey, get all tangled up. They actually organize in a very complex way. And it's important for the virus too, because, for example, influenza, there are 8 pieces of the genetic material. And if you just let them, you know, like flow around and get all tangled up, it's really difficult to make a good infectious virus if those 8 pieces of genetic material are not bundled together, right? So there's a reason why genetic material needs to be organized very, very well. All right, I haven't talked much about the, so hold on, let's see. So here again are the component of the virus. On the outside, you have the lipid membrane and the glycoprotein, which I have not talked about. And those glycoproteins are the green pieces around the virus here. Inside, you have protein and DNA or RNA. And then some viruses, they also travel with luggage, right? So these luggages are usually enzymes that the virus take along with it. And what the goal is so that when a virus infects a cell, these enzymes can actually help kickstart the reproduction process a little bit early, so the virus can lay a bit, you know, low key a little bit before it recruits other cellular proteins to come and help it make more viruses. And there might be an advantage to that. If you can lay low, then it's less likely that you're going to get detected. But here, the glycoprotein, I saved it because now we're going to talk about the idea of how a virus chooses a host, right? What makes a virus infect that, and then another day it can go and infect a human, right? So there's the that's what usually happens when we have a disease outbreak like the COVID-19 right here, right? So coronavirus are usually circulating in a population of bats, and a mutation occur in the virus and allow it to jump from one species to another species, right? And so, and that depends on the glycoprotein that you're looking at here. So if you think about a lock and key model, imagine the glycoprotein on the virus as the key, and imagine on your host cells, you have a ton of proteins covering the surface of your cell, right? And some of those proteins can be a lock that can be unlocked by the viral key. When there's that unlock event happening, there's a potential for an infection, right? So for example, the flu, this flu right here, for example, let's call it an influenza that will affect the human population. This key right here can actually bind to a receptor on the surface of your cell, and that is a key lock event, and when that happens, the virus can infect the cell, right? So that's how the virus chooses a host. You need a compatibility between the virus and the host, right? And when it chooses a host, where does the infection really occur? Right. So, for example, we're thinking about the virus that I study, dengue or Zika virus. These are the viruses that are transmitted by mosquito. So the mosquito come and bite me right here, and the infection doesn't really happen right there. The infection, the mosquito bites will allow the virus to go into my bloodstream, but then those viruses don't infect the skin cell, at the bite location. They actually infect a blood cell. So they would travel through my bloodstream until they see a cell that has the lock that they can open, right? So the point I'm trying to make here is the infection is really tissue specific. So for example, hepatitis A or hepatitis B, those viruses are specific to your liver. They can only infect your liver, the cells in your liver. Flu infects the cells in your respiratory tract, right? HIV infects your blood cells specifically. So that is where the infection occurs. All right, let me check Q&A to see if there's any compared to, say, MERS, SARS, or the Spanish flu outbreak, how serious or dangerous is the current coronavirus pandemic? That is a good question, and that's something that we are continuing to monitor, right? Because we, as the last literature that I read, the rates of transmission for coronavirus is not as, not as great. No, a little bit greater than the seasonal flu. I haven't really compared it with the Spanish flu. It is definitely more contagious than MERS and SARS in the past few years, right? But we have to answer this question considering all of the measure that public health officials are implementing, right? We have the stay-at-home order, and that really dampens the rate of transmission for COVID-19. And this is a virus that we just, even though it's related to the coronavirus that we have learned about before, this is somewhat new to us. So I know my friends at UVM, in my group are still doing their modeling work every week to see how much COVID-19 is being transmitted in the state of Vermont compared to other states, and also, globally. All right, so let me see. Go back to my slide, and then we're gonna go to the next slide, and then while that's happening, I'll look at more questions, right? So now we know what a virus is. We know how it is made. Poll number 3 is a true and false. And the question is, a virus need to be endocytosed? Big word, but what it means, it needs to be taken up. It needs to be swallowed by a cell in order for it to start replication. Is that true or false? And I told you before that a virus needs to be in the cell to replicate. All right. So while you're polling, I'm gonna go back to the question. Could CRISPR be used on viruses? The short answer is yes, but it's a very complex system. What we can do is we can put the genetic material of the virus, right, onto a bigger piece of DNA, for example, and then usually it's, you can put it in a DNA of a bacteria, and then you can modify the viral gene using that platform and hope to be able to get a good virus out of that. It's easier said and done, right? As with anything in science. So if you get a virus out of that, you have to see if it behaves the way you want it to, right? So there are ways to modify the genetics of virus, and you don't have to use CRISPR for that. We have a lot of molecular genetic tools that will help. Let's see. All right, so let's come back, and then I'll look at questions again. So a virus needs to be endocytosed. So you agree that it needs to be taken up. That's a reasonable answer, but really, technically, some virus doesn't need to be taken inside a cell. Some virus can just sit on the cell surface and inject its genetic material inside a cell, and that's all it needs to establish an infection, right? So now we're gonna look at the viral life cycle, what happens when the virus get inside the cell. Okay, so here is a very nice diagram of pictogram I found, right? So here you have the virus with its keys. Those are the yellow knobs. And then the cell locks are the purple proteins right here. There's that lock and key opening up, and that will allow the cell to form a pouch, and that can wrap itself up around the virus, right? And that way the virus is now pulled inside the cell. And then what you have is viral fusion and disassembly. And what that means is that the coding of the virus is gonna open up. And when that happens, it can release the genetic material into the cytoplasm of the cell. That's the word, cytoplasm. And that just means it's the environment inside your cell. When the viral DNA or RNA is inside a cell, it can be made into protein, right? To need protein as building block to build new viruses. And then the genetic material is also replicated. So at the end, you're gonna have a ton of new viral protein and a ton of viral RNA in this case. And those components are gonna assemble together, and then you get an immature virus, right? So this is the precursor, like, of a new virus. And as it travels, or the technical term is being trafficked through this system right here back to the surface of the cell. And as it goes through that system, the virus become mature, right? And at the end, it gets released from the infected cell. Some virus can mature outside the cell too. And by that reasoning, a lot of people say, oh, it doesn't need to be in the cell to become a real mature infectious virus. It can do that outside the cell. Therefore, we can consider it a live organism, right? And as you can see, the immature virus has all of these blue knobs on it. And then the pair of scissors, imagine that's an enzyme that will cleave off all of those blue knobs. And now you have, again, a yellow infectious virus, right? So let's recount our step there, right? You have attachment, endocytosis, uncoding, protein and genome production, assembly, and then release. All right. Okay, poll number 4. So now we're sort of like stepping outside of virology and walking into, the human system to see how virus creates disease in human for today. So my question is, disease symptoms caused by viral infections are the result of a viral product and new virion. New virions is just another word for virus particles. Disease symptoms is also caused by immune response. That's your B. And then C, all of the above.
Okay, the poll's been launched. Let's see what you think.
Okay, so I see I'm looking at your questions now, and what is your opinion on whether a virus is alive? It changes, but I am inclined all the time to say that it is alive. Yeah, all right. Does getting a virus change your body DNA permanently? Yes and no. Some virus will get clear from your system and you see no trace of it, except for an immune response generated from that virus. Some virus can actually integrate itself into your genome, but not all over your body, only specific cell type. And one of those viruses is the chicken pox. Chicken pox integrates its genetic material into your cell. And so, and it just lays there, like quietly, very low key. And once in a while, you have what's shingles, right? Okay. All right. All of the above is the right answer. And I also agree with a higher majority of people choosing immune response. Okay, and let me show you why. Okay, so, right, let's look at this pictogram. So what I'm showing you is a person experiencing a ton of symptoms, right? And this is from dengue infection. So dengue, again, is a virus transmitted by mosquitoes. Usually dengue is in the tropical area of the world. And when you get dengue, you get a very specific type of body pain and muscle pain, and usually is referred to as bone break or break bone disease. That just means that, like, if you feel like your bone has been, snapped, you get eye pain, a lot of headache, you get a rash all over your body. So it's really unpleasant. And then in severe cases, you have hemorrhagic fever. Your lung is filled up with fluid because your vasculature is now compromised. And so that's dengue, right? What happens to your body when you first get infected with dengue, right? So the bear that you see right there, that's a blood cell. This blood cell is the target for dengue infection. So when dengue infect this guy is called a monocyte, and that's a type of white blood cell, right? Dengue gets in, it will make. So what happens is dengue gets in the cell, it will start reproduction, right? So now you have a ton of dengue within that cell, and the cell recognizes that, and it goes into an emergency mode. And when that happens, the cells makes a ton of proteins, and those are called cytokines, right? So cytokines are just a way for the cells to communicate with one another, right? So, so for example, I have dengue right here. That's my mosquito bite. The monocyte in this area is now making a lot of cytokines, and these cytokines are gonna diffuse and travel through my bloodstream to like all over my body, and that can trigger other blood cell to respond to that emergency, right? And one way that they do that is to make more cytokines, right? So much that sometimes people refer to that as the cytokine storm. You know, is a fun word. It's a very fancy word to say, but it's a very serious condition, because in that stage, your body is in constant inflammation. You get really, really bad fever, and some people can, you know, die from that. So the point I'm trying to make for this slide is that cytokines is one way that the immune response react to viral infection, and that might lead to the symptoms of the disease. Okay, let me check Q&A. Where does your research take place, and how do you store viruses are stored in a freezer, -80 degree, very cold, because we know that cold low temperature can preserve the structural and the function of the virus. All right, do you see anything in the chat box, Lauren, that I need to answer?
No, all the questions are coming into the Q&A as they should. Good job, guys.
Bacteriophages be used in treatment of bacteria infection. Interesting idea. I'm not quite sure, but I can imagine that's possible. But I don't think we have that many bacteriophages. The one that we work in the lab, you know, usually target very common bacteria like E. coli. I don't know if, you know, there's bacteriophage for like Salmonella or Listeria, you know. But at the same time, bacterial infections aren't as common now as they are, you know, decades ago, because we have very good sanitation and hygiene practice these days. So bacterial infections aren't, you know, on the top of the list. So they don't really have that much attention, which is unfortunate, because bacteria are fascinating to study. All right. What are some example of diseases caused by viral infections? I can say disease caused by viral infection. So for example, dengue causes hemorrhagic fever. Zika infection can cause congenital syndrome like microcephaly. You know, when you see babies with like tiny head during the Zika outbreak, that causes that. HIV is interesting because what it does, it weakens your immune system, and that makes you susceptible to many other infectious diseases. And then there's a lot, you know, there's the flu that causes the seasonal flu, etc. All right, let's move on. And then I'm gonna go back to more questions later. Okay, so now we started talking about the immune response, which is the focus of my study, right? And it's an interesting topic and it's very complex, right? But if you go to a immunology class, the first thing that you learn is the layers of immunity or of the layers of defense that we have to protect ourselves. And if you can tell me, like, take a guess and put it in the chat box to see, like what you think is the first layer of defense that we have. And then I think Lauren can help me.
Yeah, so some people are right, and the first layer is skin.
Good job. All right, what else besides skin?
Nose hair.
That's also a very good idea. Yeah. So like
isolation.
Well, the biological defense, right? Like a few weeks back, I was like, my nose hairs are getting too long. It's getting itchy. Should I trim it? Because if
we
I
got
trim
mucus and macrophage, T cells, surface proteins and oils.
So, good. So the first layer is actually the physical barrier, so your skin and your mucosal layer, right? So like the fluid that you have in your nostril, right, in your mouth. And so those are the layers that are designed by our body to slow down the movement of microbial pathogen, right? So because think about your booger, it's quite thick, right? Because it's so thick. Bacteria have a really hard time moving through it to get to the cells that they want to attack, right? So that's your physical barrier. And then when you go zoom all the way down to the cellular layer, then we have two arms of the immune system. And the first arm is the innate immune response. Oh, my ends are funky. Okay.
I was gonna let you know it's a mirror image, so everything.
Yeah, because I was trying to mirror image because of the Zoom thing. Anyway, so the innate immune response, and the way to think about the innate immune response is, it's natural, it's general, right? So for example, if I have someone break into my house and I'm sitting in the kitchen, I'm gonna defend myself by throwing, whatever I have around me at that person. I can throw them a muffin, I can throw my chicken at them, right? So those are not really weapons, those are food, right? So this is sort of like the innate immune system is like an emergency response that we're gonna throw whatever we have at the invader, right? And in that case, we have these cells right here: the basophil, the neutrophil, the eosinophil, and they have a lot of you can see they have a lot of these little spots in them. And what those are actually packets of chemicals, right? They could be the cytokines. So when there's an invader, these cells are going to release those cytokines and those chemicals into the bloodstream, and that sets up the innate immune response. And sometimes it's strong enough to clear the infection, sometimes it's not, right? So that's the major part of the innate immune response. And you have someone mentioned macrophages. These are macrophages. And the reason why they're depicted this way is because their job is to roam around in your bloodstream, and they take a sample here, a sample there, right? And if it happens that they sample a bacterium or a virus, then they will also set up an immune response by releasing their cytokines, etc. Right? Dendritic cells also does that. They will also go and sample and engulf, invaders and other molecules. But what's really cool is when they do that, they will chop up whatever they swallow. They will digest that, and then they throw it up and they hold those little fragments, if you will, of viruses. So if they eat a virus, they're gonna chop up that virus, and then they're gonna present the viral fragments. Say, I ate this, you know, and now I'm presenting the viral fragment to you, and um, and they are really cool because they bridge the innate immune system with the adaptive immune system. Adaptive immune system, right? And the adaptive system is consisted of these T cells, CD4 T cell, CD8 T cell. You usually hear about those. You have regulatory T cell and B cells. So these dendritic cells are gonna show this viral fragment to these T cell and say, hey, look at this. It's from a virus, I think. And these T cells are gonna recognize that fragment and say, oh, that doesn't belong here. That must come from a viral invader or a bacterial invader. So now we need to start working, right? So now you see this T cell is dressed as a superhero. And what it does, it goes and it helps, right? Usually they're referred to as helper T cell because they go and they help. And who they help is B cell, right? So CD4 T cell is gonna come and nudge B cell, say, hey, we have an invader, do something, right? And what B cells does is that you see all of these molecules on the outside of the B cell, those are antibodies. We've heard a lot about antibodies lately, right? So antibodies are simply molecules that can bind to the virus, right? We'll talk about B cell later. So when T cell go and activates B cells are gonna start releasing those antibodies, right? And then you have this CD8 T cell here who dresses as a ninja. What it is also called cytotoxic T cell. What it does, it kills infected cells. So when, say, this T cell see this dendritic cells with that viral fragment, it's going to kill this dendritic cell. The advantage of that is if there is viral replication happening in this dendritic cell, then killing it will stop the viral reproduction cycle. Therefore, you will lower the amount of virus in your system, right? All right. So before we move on to the next question, have a good look at all of these for a moment while I'm looking at your questions. Can COVID-19 end before we have a cure? If we, maybe not. It takes a long time to come up with a cure. There's talk about, you know, how to safely reopen society, and that's the topic of debate. I'm not sure, you know, we're doing really well as a state to control the transmission of the disease. But, you know, if we are not able to control people coming in and out of our states or in and out of the country, then it might be a very long period before it really goes away, all right? Is it possible to engineer a virus similar to a zombie virus that targets the brain and disables things like reaction to pain and proper functioning of the nervous system? Very good question. We know that some infection by parasite can sort of alter the brain of animal. And I forgive me for this, but when a mouse is infected with Toxoplasma gondii, which is a parasite, this mouse infected will then go seek out cat urine. They become attracted to urine of a cat. Why? Because this mouse is now carrying the parasite, and this cat is going to eat this mouse and therefore become infected with parasite. And this parasite needs to be in the cat in order to go through its life cycle completely. So yes, there are ways to, well, it's it's far-fetched, but there may be ways to alter, how the brain function upon an infection. But the tricky part is to translate what we learn in an animal model to a human model, because we're very different species, and there's a lot of ethical issue regarding testing on human. So it's a really interesting idea. There's a lot of discussion that should come out of it, but to really get there, it's gonna take a long time. All right, so now I'm gonna go to my next poll, which is regarding vaccine, right? So we've been talking about getting a COVID vaccine. And the question is, which components of the immune system is engaged or should be engaged to build long-term immunity when a person is vaccinated? So what I didn't tell you, I think I did not tell you, is that the adaptive immune response is engaged, is thought of as, the memory component when you become infected with something, with a virus, with a bacteria, right? So here we're trying to build long-term immunity with vaccination. So which of these components do you want to engage in order to build durable long-term immunity?
All right, the poll's been launched. And if you, for some reason, don't see it, write in the chat box. But it looks like most of you are seeing the polls today.
All right, so I see a question that's been there for a little bit long. What's the difference between viruses and bacteria? They are simply different organisms, right? I think one, we know bacteria are alive. We're still debating whether virus is alive or not, right? Bacteria can function, most of them can function outside of a host cell. Virus can't, right? So those are two differences. Size, bacteria are usually, magnitudes bigger than viruses. Bacteria genetics is much more complex. Bacteria genomes can have thousands to hundred thousands of genes. Virus genome usually contain about, handful, you can count on 10, 20 genes, right? So I would say virus is much more evolutionary advanced compared to bacteria, right? Because it can propagate, it can reproduce with very minimal instruction, where bacteria need a lot more. All right, so those are some of the differences. How easy is it to engineer virus? Do we know of any engineered virus? It's relatively easy to mess with the genetics of a virus. I'm not sure if you've heard, but a few years ago, I forgot where it is, but a lab was able to engineer an influenza virus that allow it to be super easy to aerosolize, and that causes a massive problem, right? Because flu is transmitted through the air, and if it's so easily aerosolized, that it causes a problem, right? So that's an issue with engineering virus. So that's one example. We have the Zika and Dengue virus that I'm working with. Some of those are engineered as well. So we go in, we modified it a little bit to make it weaker, and we can try to use that weakened virus as a vaccine candidate, right? It still needs to be tested and all that, but we can try to make it weaker. So that's an example. Okay. All right, let's go back. So the polls say you need to engage all of these components to make sure that you have a good immunity, which I agree. right? In the past, adaptive immune response is also a very good good option for the last 60 to 100 years or so. We always think about immunity in terms of B cells and the antibodies that they produce. Uh, Only recently we also know that T cell also does a lot of the work, right? So it will be good if we can engage both T cell and B cell, because if you think about it, T cell go and help B cells, right? So if you can, get everyone involved, that will be really good. For the people who chooses innate immune response, I'm not sure what your rationale is, but some vaccine work better if you engage the innate immune response as well, right? So you need that like first emergency, status in order to set up a really good immune response for later on. So um, if you can't do anything, then you engage B cell. If you can like engineer your vaccine a little bit more, then it will be good to get T cell and innate immune response involved as well. Good job, everyone. All right. So now, how does the body fight infection? So let's go back to the scenario where I have my blood cell infected with dengue, right? So they create a bunch of these cytokines, and then they release these cytokines into the bloodstream. And some of these cytokines actually come and bind to B cells, right? And the B cells now will know that, oh, this is an emergency signal, so I need to start working, right? And as I told you earlier, that B cells, its job is to make antibodies, right? So now this B cell is throwing all of these antibodies out, and these antibodies are going to bind to the virus. Several things can happen. They can cover the entire surface of the virus, and now so the keys aren't available for the virus to go and unlock other cells anymore, right? So that's one way you win by number, right? You just cover these viruses and you gum them up, right? And then eventually the virus gets degraded by, proteins and other factors in your blood. Another way, in the case of bacterial infection, these antibodies can actually bind to the bacterium, and a bunch of them are going to bind to the bugs, and eventually set up a signal for protein to come and put holes on the bacteria, right? And when that happens, you can just live with holes on your body all over. The bacteria will die, right? So that's like several ways that antibodies can prevent infection by or fight the infection by preventing these microbes from infecting other cells, right? So the point here is B cell making antibodies, right? So we've heard recently in the past few weeks about, the antibody test for COVID-19, and the people who recover from COVID, we think that they have really good antibodies that can defeat the coronavirus. So that's why people are asking to donate their blood, right? So in the blood, you can extract the antibodies out and use that antibodies to treat people who are not able to fight COVID alone, right? So that's one way to sort of like help combat COVID-19, is to use antibodies. And in our lab, we are also working with antibodies that can neutralize or inactivate Zika virus or dengue virus. And if you develop that, enough, you can use that as an injection. So when a person has a mosquito bite and say, oh, I think I have Zika, you can just give them a shot of the antibodies, and that will help clear the virus. And that's the thing that we're trying to do with COVID-19 right now too. It just takes a long time to come up with a good antibody against the virus. All right. Question. Oh, all right, let's go to the next slide. Right, so now when we think about long-term immunity, we think about memory, right? So what I'm showing you here is how B cells develop, right? B cell is an umbrella term for all of these cell types. So you have the plasmablast, which are the cells that show up really early after the infection. So in COVID-19 patient, B cell shows up between 7 and 14 days, right? And after that, they will turn into different cell type, right? So plasmablasts can become memory B cells, right? So now they have a memory against COVID-19. And what memory B cells do is they circulate in the bloodstream with all of these antibodies now that can recognize COVID-19. And when the person is infected again, we think that these antibodies are going to bind to the virus, and then the cell can go to work again, and they will repeat the cycle. They become plasmablasts, and then they'll make more memory B cells and more long-lived plasma cells. Now, long-lived plasma cells are also B cells, right? What they do is they go and live in your bone marrow, and they call long-lived because they live for decades. They can live for like 30 years, 60 years, really long time. And what they do is they make antibodies again, right? Antibodies is so important. And these antibodies will then flow out into your bloodstream and circulate in case you have another infection, and the antibodies can quickly gum up the virus and then clear the infection, right? So that's how you recover. All right, now before moving on, let me look at the questions. So I answered the zombie virus. Recent popular Japanese show called Cells at Work. How accurate is it? I haven't seen the, the series, so I'm not quite sure. All right, why did you want to study viruses? I don't quite study virus. I study the immune response to virus rather. But the thing a lot of people like about virus is how transient they are, and you know they are very small and they can do so much damage. You know, so if you can figure out what gives them so much strength, then you may be able to improve our life a lot. Recently, someone recently read that the first patient was cured of HIV. How did the cure work? I'm not familiar with the study, but I know that we have really good therapeutics. We have really good drugs for HIV now. We're still working on a vaccine, but treatment is really, really good. And usually the way that they measure HIV drugs effectiveness is to see if they can detect the virus genetics in your blood, right? And so if they can't see it in a blood test, then it's assumed that you'll have the disease under control. There's several things that can, sort of like mess with the result if your machine is not sensitive enough to pick up, little bit, just like a tiny amount of viral material, then, it will say negative, but really there might be a little bit that is lower than you can detect. But to go back to your question, usually for viral disease, if you are able to reduce the amount of viral material in your blood, then that's a positive sign, right? So for example, for COVID-19, people were arguing whether hydroxychloroquine were effective, and some study give the drugs and look at how much viral genome that they can detect, and there have been conflicting results. So that's one way to measure how the drugs really work. Okay. Lula Gage asked, if you have had the same sickness several times, why isn't your body immune to it yet? I thought that's how vaccine works. And really, that's how vaccine is supposed to work. The reason is why you might not have immunity is because viruses are constantly changing, right? It changes quite a lot. And it's because of the type of genetic material that virus has. Um, and also because it replicates so much, right? Every replication, there's always a little bit, some amount of mistake. You know, when you make a copy, in making making copies of genetic material, there's always some level of mistake, and maybe that mistake leads to the formation of a different strain of the virus, right? That's the issue with the flu virus. It changes every year, so that you're never really gonna get good immunity against all seasonal flu, right? And that's the reason why right now we're trying to work on a universal flu vaccine, so you can just get one shot and be good for life. But that's really hard. And the bottom line is the virus changes so much that it's really difficult, you know, for the most part, to build a good vaccine against all the virus. All right. Okay, so now that we've talked about vaccine, and I want to, let's see, ask you, how long do you think it takes to produce a vaccine or a drug? Can we come up with a COVID vaccine before we let everyone out of their house?
All right, I just launched that poll. How long do you think it takes?
All right, I see 5 to 10 years. Reasonable, but very optimistic.
Anyway, I'm just gonna have you watch the time.
Okay, yep. Yeah, all right. So, okay. So I see that most people say 5 to 10 years. Then that's reasonable. Um, so it really varies, right? It depends on the disease type. Does a lot of people have it? But usually it takes about, 10 to 20 years for something to be, approved and come out for public consumption. And the reason for that is because drug and vaccine development is a very laborious process. So what you have to do is to have preclinical data, and these are data performed on animal models that are not humans. So you start with a mouse model, and then you move on to, say, a rabbit model, a monkey model. And when you show that the drugs are safe in these animals and they are effective, only then can you move the study into the human population. And we have to be super careful with human trial, right? So you start with a very small number of participants, just a few tens, right? Less than 100. And the only thing that we want to see here is whether the drug or the vaccine is safe when we give it to people, right? So if it is safe, we can move on to phase 2, where we give it to several hundred people. And at this point, we're still going to need to know that it is still safe, right? And if it is safe, is it also effective? Is it effective? Does it work on 50% of people, 30% of people, 90% of people? If that percentage is too low, it goes back to the, phase 1, and you need to modify your study or modify the drug. But if it is safe and effective in phase 2, you move to phase 3, several thousand people. And so, for example, the vaccine that we are working on right now at UVM for dengue hemorrhagic fever, we started that work in like the 1990s, right? So what? That's like 30 years ago. And only now it is in phase 3 in Brazil. And phase 3 itself is taking 5 to 6 years, and we haven't gotten the data back yet, right? And if it all goes well, if it doesn't, make the disease worse, if it is effective, then you move on to FDA review, and if the FDA approves it, then you can get it made by a manufacturing company, and you can start selling it to the public. So it takes a long time, and so in terms of COVID-19, it's really hard to say that we're going to come up with a vaccine, in a few months, or a drug in a few months. So, you know, we're still operating by very conservative, numbers, trying to control the spread of the disease rather than, hoping for a vaccine to come out or a drug to come out and cure everything, right? So phase 4 is not really crucial, but if you have a similar drug, you can go to phase 4 and see, oh, if this drug is, better or, not as effective, then that's a comparative study. But if you pass phase 3, the FDA reviews it, approves it, and then you're good to go. All right, so I think that's the end of my talk. Here's again, to remind you the complexity of the immune response or the cell type. Here are the several resources that I pull from when I made this PowerPoint. So feel free to go and play around. They have some really interesting things to look at, and some really nice read. All right.
And so before we go to questions, I'm going to launch one last poll. I'm just going to ask for some feedback from everybody before we get to the questions, because I know some folks might need to hop off. So I'm going to launch this poll. Just let us know what you think about today's, just the overall cafe. And then we would love to know if you learned anything. So I'm launching that poll. And while that poll is going, Huy can start looking at the questions to decide what he's going to answer next. You also can write any comments you want in the chat, but it's best if you also take this poll. I do want to say we have a great lineup of cafes for the month of May, and we're scheduling some into June already. So we'll send that link out to you because we definitely have some really good ones coming up. So keep taking this poll, and then we're going to get to all those questions that are in there. And we know some of you will have to jump off, so that's okay. And there's some very nice thank yous. I'm gonna put the chat where you guys can, I turned it off because there was too much distraction, but if you need to say goodbye to some people. And wait, why don't you start in with the questions that are there?
All right, so would you recommend going to UVM if someone is interested in biotechnology and immunology? And there are lots of research opportunities at UVM? Absolutely. UVM is one of the best university in our country in terms of being in the front line of research, right? So we have research from, let's see, my group is immunology, so we learn a lot of vaccine. We learn a lot about how the human body responds to infection, and if the vaccine is effective, that sort of thing. We have labs that focus specifically on the biology of virus, right? So you can figure out why this virus is effective in transmitting from one species to another. We have a good lung program, so biology of the lung. We have a really big cancer center. So there are a lot of opportunities for research, interesting research at UVM. You can go from, single molecule, so study one protein, and you can go across the spectrum to learn about translational disease or translational science, where there's more applicability, between your research and the patient's health. So I do recommend UVM. In terms of biotechnology, I think we are a little bit behind, but there are, people pushing for our research to be more translational, right? So we don't want to just like hanging out in our lab. We want our research to have a practical impact on people's health. So there's a push for that. I definitely recommend UVM, not just because I go there. We also have a lot of funding. UVM is free. I would say, we're very well supported. All right. Could COVID-19 change every year due to mutation, similar to the flu virus? Potentially, maybe. I mean, this is the first year we have it, so I would hate to say that, oh, let's wait for it to come back next year to see it has mutated. But yeah, viruses are prone to mutation, and so there's always a chance that it can come back, and we're not gonna be equipped to deal with it. Is there a way to test drugs without using animal? Unfortunately, no. Right now, the common way to do it, to test drugs, is to start with an animal model, because you have to make a gradual transition from a petri dish to a human, right? So jumping from a petri dish to a human is just too hasty. So you need to go through mouse and then eventually monkey. I know it raises a lot of ethical issue and financial issue for many institutions, but that's the best way we know. All right. Were there cases when a bad vaccine got through the process? If someone bribed the FDA, turned out to be some negative effects. I don't think people do that intentionally, because we're dealing with, human health here and people's well-being. I don't think people, I believe in general that people are good, right? But there are vaccines that perform really well in small study, but in a real population, it's a little bit more problematic. One dengue vaccine is not the one that we worked on, but one that was approved a couple of years ago is now causing a lot of problem in the Philippines, right? And that's not because of the bribery at the FDA. It's just because we just don't know enough about this virus. And from the data that we see, it works, but in reality, it doesn't work. So that's in phase 3, and beyond. And so what we need to go back is to go back and to sort of like modify the recommendation. Maybe the vaccine only works well for people from age 9 to 45, right? So there are cases that drugs, cause problem, even though it looks good on paper. Do elderberries really improve the immune system? So I've heard. I have not tried, and I cannot attest to that. Could COVID-19 change? Yep, we answered that. Will some of these steps be rushed due to circumstances for COVID vaccine? Absolutely, yes. So if it's an emergency, you can push it through the process rather quickly. Can a vaccine act on several viruses at once? Not likely, because viruses are very different and vaccines are very specific to the virus that it's designed for, so not likely. If a plant got COVID-19, would it be affected? I don't think so. COVID-19 affects cells of the lung, and plants don't have lungs. And if it is affected, I wouldn't know, because no, I don't think so. Could we get virus from an animal? Yes. The coronavirus, other versions of it, is known to come from bats, and it's transmitted through different kinds of intermediary animals. So I think MERS came from bat and then get transmitted through camel. SARS came from bat and then is transmitted through a civet cat. Yeah, could an animal get COVID-19? I don't think so. Well, COVID-19 is the name of the disease, but an animal can definitely get coronavirus, right? But it won't manifest as a disease like it does in human. All right, I answered all the questions.
Wow, that was a lot. Can you unshare your screen? And I'm just going to throw one last slide up. For those of you, we still have a great number of you still here. So I just want to share with you all that are still with us our final, this is the list of upcoming cafes. And you can see the registration link there. You go there and you're gonna have to register for each cafe separately because we have different Zoom links every single time. And you can see that we have lots of other programs. But I would love for you all to take the time right now in the chat to thank Huy for giving us so much wonderful information today. Thank you for coming and telling us all about this. And your graphics were so enjoyable to look at. Who knew that cells were such happy little creatures?
Yeah,
so thank you all for joining us today. And we hope that we see you again. Just take your mouse and you can click end meeting to leave.